Has that worked all right, John?
It's great.
Perfect. Okay. Kia ora koutou, ko Joe Carpenter tōku Whenua. For this talk, I'm going to be talking about the impacts of possums and rats on forest fruit abundance. This is work that we did in partnership with the Tūhoe Tuawhenua Trust, and this research was really initiated by Tūhoe Tuawhenua mā Tauranga that was collected over many years, especially by Phil Liver. So I'd just like to really thank Phil and all the elders that contributed that knowledge, because this research wouldn't have been possible without it. So at the beginning of the More Birds program, we had several hui with the Trust to discuss and co-design research that aligned with the Trust's interests and the theme of More Birds, and was guided by Tuawhenua mā Tauranga. And one thing that really emerged from those conversations was that restoring kiraru to their previous abundances in Tuawhenua forest was critically important to the Trust. So we needed to know what limits kiraru. We did a review of the national scientific literature, and what we found, like John mentioned in the earlier talk, was that predation by introduced mammals was the major limiting factor for kiraru in intact forests, but that forest quality was also really important because kiraru do breed in response to fruit availability. Of course, mammalian predators tend to determine the outcome of those breeding attempts though. So Tuawhenua elders had observed changes in their forests over time in terms of forest quality. They seemed less resource rich. There was less fruit around, and some of the fruit appeared smaller as well. There were several potential mechanisms that were proposed to be behind these changes. The first was the logging of podocarps that happened in Tuawhenua forests between 1950 and 1975. The second was the impact of introduced mammals, the impact of possums and rats on fruit abundance, but also the impacts of ungulate causing changes to forest structure. And finally, there was also a potential impact of changing climate, of the forest getting warmer and drier. So we co-designed a couple of different studies looking at these various mechanisms, but today I'm going to be predominantly talking about our research that looked at the impacts of possums and rats on fruit abundance. So how do possums and shiprats impact fruit abundance both separately and together? We can become so focused on the predatory impacts of these two species that it's easy to forget that fruit is a really favoured resource for both possums and shiprats. And Tuawhenua Kaumatua had noticed a reduction in fruit production for some plant species, which they thought could have been caused by possums browsing flowers, new shoots, and the fruit itself. There's been a couple of studies that have shown possums affecting fruit abundance for a couple of plant species. However, no studies had tried to separate the effect of possums and rats on fruit abundance. Why is this important? Well, when we do aerial control regimes, these often can keep possums down for quite long time spans, up to potentially 6 years, but they only briefly suppress rats. If possums are the main culprit suppressing fruit and flower abundance, then this type of regime is likely to still have positive outcomes in terms of resource availability for birds. But if rats simply fill the niche of the suppressed possums, then those benefits could be negligible. So there's a couple of mechanisms by which possums and rats could be affecting fruit abundance. The most obvious is this kind of direct consumption of large favoured fruits, such as tawa or hino. But a less obvious mechanism, which is more specific to possums, would be the consumption of foliage or flowers of the plant that then impacts the plant's ability to produce those large fruit crops in the first place. So this is a much more cryptic, insidious mechanism. So we co-designed a study with the Trust which used a before-after control impact design across 8 25-hectare blocks within Tuawhenua Forest. We collected seed fall monthly in seed traps, and every 6 months we indexed rats and possums using tracking tunnels and chew cards, respectively. After almost 2 years of this baseline monitoring, we began pest control treatments. So on 2 blocks, we controlled only possums. On 2 blocks, we controlled only rats. 2 blocks, had had both controlled, and then we had 2 non-treated blocks. And I'd just like to really thank Slane and Pato. Pato is just pictured there, who did so much of this mahi. We're incredibly grateful for all their hard effort in the forest. So here's just a quick map showing where these blocks were situated across the Tuawhenua region. Tuawhenua is the central hinterlands of Te Urewera. So you can see from the various colours that those are the different kind of treatments the blocks had. And here we are at the beginning of the study establishing the seed traps. So there were 30 of these seed traps established on every block. 20 were set up in the middle of the block, just in a grid formation. So they were intended to collect forest level seedfall. And then we also specifically targeted 10 traps beneath female miro, or as they're known in Tuawhenua, toro miro trees. The reason we specifically wanted to monitor toro miro fruit production was because it had been, that species had been really identified as really important for keridu and keridu breeding. So we particularly wanted to understand potential impacts of pest mammals on that species. So we'll just start by looking at the residual pest abundance results. So in these graphs on the y-axis, we've got the mean percentage tracking by rats and the mean percentage of chew cards chewed by possums. It's there on the y. The graph in the red is the ship rat tracking rate. In the yellowy colour is the possum chew card index. The 4 panels represent the 4 different treatments, and we have time on the x-axis. This black line in the centre represents when we began our pest control treatments. So the first thing I would like to point out to you is that the pest control treatments were generally successful. If you look down at the rat control treatment, for example, we did most of the time manage to suppress rats below 5%. Our possum control treatment as well may not look like the biggest reduction in possums, but this is statistically significant. And this particular index of using a one-night chew card survey for possums isn't very sensitive. So we also have to keep that in mind, that it was a little bit hard to detect the reduction as well. You might notice that at the very end, it looks like things go a little bit haywire. That's just because our last pest monitoring survey happened a bit late because of weather. So it actually happened a couple of months after the study had finished and we had finished pest control, which is why the pests obviously rapidly shot back up to really high levels. The second thing I'd like to point out is if we look at this panel, is that our possum control treatment resulted in increased rat tracking. So you can see when we begin pest control with the black line, our rat tracking goes up. If you're comparing it to the plot above, which is the non-treatment, it may not look like it goes up that much, but it's important to also look at this before period where it appears that these possum control blocks were originally perhaps a little bit less ratty than our non-treatments. But then once we start pest control, they become as ratty, if not rattier. Ratty is the technical term, of course. So this wasn't an entirely surprising result. It's been shown by Wendy Rusco and others that often when you do possum control, you end up with more ship rats. Possums compete with ship rats and tend to be thought of as the dominant competitor. But what was more surprising is that we also saw the inverse result. So when we did rat control, we also ended up with higher rates of possum detection, more chew cards being chewed by possums. This was surprising, but there are some suggestions that this competition between possums and rats could be quite density dependent. So depending on what the density of rats and possums are, at some stages rats actually could be able to compete for food resources more successfully than possums. So potentially our removal of rats may have allowed more food resources to be available for possums. I guess the overall picture I'd really like you to take home from these plots is that, as we know now, any kind of pest control manipulation, which just removes one pest mammal in a suite of pest mammals, will always have these cascading effects to the other mammals within the system. So we're not just dealing with a possum control treatment with less possums, we're dealing with a possum control treatment that also then has more rats. So it's important just to keep that in mind as we go forward into the seed rain results as well. So overall, we captured almost 80,000 seeds in our seed traps from 24 different plant species. The 5 most frequently captured species were rimu, toromiro, hino, Leucopogon fasciculatus, which is soft mingi, and various species of pseudopanax. I'd just like to point out that counting and identifying almost 80,000 seeds is an enormous effort. It takes months and months of sorting and drying and checking. And on top of that, we also, these seeds were scored on whether they'd been eaten by mammals or dispersed by a bird or eaten by an invertebrate. So it was a lot of processing. And with that caveat in mind, we haven't quite finished processing the data. So for that reason, I'm going to show you some raw data plots, but we haven't got onto the analysis or doing proper statistics on them. So just a little disclaimer. So let's start by looking at the overall seed rain results. This is for all the species, the plant species combined. So on our y-axis in this plot, we've got the mean seeds per trap, all species summed. On the x-axis, we have time. Again, that black line represents when we begin the pest control treatment, and the various panels are the different types of treatment that were imposed. So what you are looking for in these plots is obviously we have a before and an after session, and we're using the before to create a baseline to understand the intrinsic variation that will just be inherent to these sites and to these plant species. And so what we're really looking for is we keep the before in mind, but then we look at the after, and we are looking for whether any of the treatments seem to have increased the fruit production compared to the non-treatment. So for example, I think looking at these plots, it looks like potentially our possum control treatment has increased the amount of seed rain available compared to the non-treatment when we look at all these species combined. If we move on to look at rimu, again, this is an interesting example because rimu is obviously a masting species. It's incredibly variable from year to year, and we can see that in the first 2 years of the study, it didn't really do much at all, but it luckily seemed to have a big year in 2023 at least. And when we look at that, it does look like our possum control treatment resulted potentially in more rimu fruit. Obviously, all these plant species are going to have different palatability to possums and rats. Rimu fruit is very popular with both species, but the foliage is not eaten by possums. So this is a species where we would expect the impact of possums and rats to be quite direct. It's mainly just driven by that direct consumption of the fruit or seed. Moving on to Hino, in this particular plot, it looks like potentially our treatment, which was both possums and rats controlled, may have resulted in more Hino fruit production over time. Hino fruit, again, is really popular with both rats and possums do also eat Hino flowers, but not the foliage. So this is a species where we could have both direct impacts of possums and rats on the fruit and indirect impacts of possums eating flowers and generally suppressing the ability of the trees to produce more fruit as well. Coprosma, this is a real mixed bag. Obviously, the coprosmas just did a lot more in the before period for some reason and not very much in the after period. Coprosmas obviously encompass a raft of species, and these species are going to have different palatability to possums and rats. For example, possums really like Coprosma robusta fruits, but the little twiggy coprosmas such as rhamnoides are probably a lot less favoured. And the same with rats. Some will be favoured and some won't be. Another thing to keep in mind with these coprosma results is that most of these seeds we actually collected had been dispersed by birds. So there's also this, I guess, kind of cloud of variation overlaid these results as well, that they've already gone through birds. If we turn to miro or toromiro, which was obviously a species we particularly wanted to look at, This fruit is very favoured by rats and somewhat by possums. Rats eat the seed and possums eat the flesh. The foliage is not eaten by possums, so this is a more direct impact. But there's not really an obvious difference, I think, between the pest control treatments and how it affected meadow. But another thing we can actually look at is we can look at the proportion of seeds we collected that had actually been chewed by mammals, by possums or rats. So in this graph, what we're looking at instead is on the y-axis, the proportion of the crop we captured, which showed signs of mammal damage. And the seed shown down here is a rat-chewed meadow seed. A very ancient one actually, but they look the same even if it's a contemporary seed. So in these plots, I think we can see that when we are doing possum control and also the non-treatment, we end up with between 60 and 80% of the captured meadow fruit being eaten by mammals. But it looks like when we control both possums and rats, or just rats, we capture most fruit intact. It hasn't been chewed by mammals. So yeah, there's a potential direct effect on the meadow fruit itself. So what do we take home from this? I think the most obvious thing probably is that seed rain data are really messy. We're dealing with a soup of all these different factors that can affect seed rain, especially when you're looking at it from a community level. We're still to run these statistics, and I think once we do, we are going to find some significant effects of our pest control treatments on fruit production. But on the whole, I'd say we haven't detected a huge impact of controlling these mammals on fruitful. It's more subtle. And I don't think this means that mammals aren't important. It may just be for some plant species we didn't measure their seed rain for long enough, or in some cases, perhaps our pest control, although we know we did suppress the pests, maybe we didn't suppress them enough in some cases. Another factor that we think could be really important is the potential impact of climate change on the fruiting of some of these species. So for example, tawa is a dominant canopy species in these forests. It's over pretty much every single seed trap we have, but we captured very few tawa fruit across the 4 years of the study, even though it should be fruiting every year really in variable amounts. So what we think is happening with the tawa is that Whenua forests have become warmer and drier, and that's shown in this plot here of summer temperature at Whenua over the last 100 years. And this has also been observed by Whenua elders, that there's less frost and the forest is a lot drier than it used to be. And research done by master's student Oscar showed that these changes to climate will have been directly impacting tawa fruit production. So it could be in some cases that this impact of changing climate actually eclipses the impact of pest mammals on some of these plant species. Thank you very much.
Great. Thank you, Jo. Fabulous timing. It's 11:50. Maybe we have time for one question. Someone's asked, what poison are you using for the control? That might be presumptive that you used a poison, but how did you manage this tricky job of killing rats or possums, but not the other?
Yeah, I thought that would be a question that came. For the possums, we used leg hold traps and cyanide. And for the rats, we used bait stations with either diphathinone or double tap. We just kind of alternated between them and pulsed it. And we also used A24s. Yeah, but it was a tricky job to try and control one and not the other. And when we did the both possums and rats controlled, we threw everything at it, all those various treatments.
Yeah, tricky, very complex, and you certainly showed that complexity, in spades. Thank you very much.